Now that CRISPR gives humanity unprecedented power to make targeted changes to DNA, how can gene editing prevent disease and reduce suffering without causing irreversible biological harm, widening inequality, or allowing present generations to redesign future human
Right.
inheritance?
I mean, that is literally the central tension we are facing right now with this technology.
Yeah.
Because on one hand, CRISPR has this undeniable potential to, help treat severe genetic diseases and completely transform biological research.
Yeah.
But then that exact same programmable power, if it's misused, it could enable unsafe or unequal, ecologically disruptive or heritable interventions that change the human germline forever.
Wow. Yeah. It's a massive, I mean, it's an incredibly complicated landscape. But to answer that question, we are
Mhm.
looking at the seminal book, A Crack in Creation by Jennifer A. Doudna and Samuel H. Sternberg.
Mhm.
And our mission in this deep dive is to basically figure out where this technology came from,
Hmm.
how it actually works, and, who gets to control it. So to do that, we're going to explain CRISPR's bacterial origins, the mechanics of guide RNA, Cas9, DNA cutting, and cellular repair. And we'll explore its therapeutic applications, its very real scientific limitations, and draw a hard line between somatic and heritable editing.
Right, which is so important.
Yeah, and we aren't just looking at humans. We will look at gene drives, issues of inequality, disability perspectives, and finally, how global governance is trying to manage all of this.
It is such a wild journey. I mean, we are tracing a path from literally microscopic defense mechanisms in bacteria all the way up to international policy
Mhm.
and basically the future of evolution itself.
Okay, let's unpack this. We have to start with where this incredible power actually came from, because as it turns out, scientists didn't just sit down in a modern lab and, dream up a genetic engineering tool from scratch on a whiteboard.
No, not at all.
They actually tapped into a billions of years old microbial defense system.
They absolutely did, yeah.
Mm,
For a long time, the origins of CRISPR were just this obscure curiosity in microbiology. Like, nobody really knew what it was for.
right.
But if you look at the microscopic world, bacteria and archaea have been fighting this invisible, um, just relentless and brutal war against viruses, specifically viruses called bacteriophages.
Okay, bacteriophages.
Yeah, and they've been doing this since the dawn of life on Earth. And these viruses outnumber bacteria 10 to 1 in some environments.
Wait, really? 10 to 1.
10 to 1.
Yeah.
It's an onslaught. So to survive this, some bacteria evolved a highly sophisticated adaptive immune system.
Hmm.
So when a virus attacks a bacterium, and by some miracle the bacterium survives, it actually captures a tiny physical snippet of the invading virus's DNA.
I love the way the authors describe this in the book. It's basically like taking a mugshot of the invader and putting it up on a biological bulletin board.
That is a great way to visualize it, yeah. The bacterium inserts that viral mugshot into its own genome, specifically into this region characterized by clustered, regularly interspaced short palindromic repeats.
Which is a mouthful.
It is a mouthful,
Yeah.
which is thankfully why we have the acronym CRISPR.
Right.
So these stored snippets of viral DNA, they're called spacer sequences. And you can think of it as a vaccination card, basically.
Oh, okay.
Yeah. So if that exact same virus ever attacks that bacterial lineage again, the bacterium transcribes those spacer sequences into RNA.
So it remembers.
It remembers that RNA acts as a biological guide. It teams up with CRISPR-associated proteins, which, we call Cas proteins.
Okay, Cas proteins. Got it.
Right. And the guide RNA physically leads the Cas protein right to the matching viral DNA that's trying to invade, and the protein just cuts it, neutralizing the threat.
Wow. So if I'm understanding the timeline of the book correctly, we didn't invent anything new here. We basically scavenged a weapon from a microscopic battlefield and figured out how to aim it.
Scavenged is exactly the right word. But, the leap from just observing that natural system to actually possessing a programmable molecular tool, I mean, that's the real miracle here.
Yeah.
Because the bacterial weapon was highly effective, but incredibly complex in its natural state. The massive breakthrough, like the real paradigm shift, happened in 2012.
Okay. What happened in 2012?
Well, researchers figured out how to take the dual RNA components of this bacterial immune system and engineer them into a single guide RNA.
Ah, okay.
And they proved that you didn't just have to use this to cut viral DNA. You could actually program this system to find and cut practically any DNA sequence you wanted.
And we really need to get the credit right here, because the history of this discovery is fascinating, and it is, often oversimplified in the media.
Oh, absolutely.
The book we are anchoring our discussion on, A Crack in Creation, is written by Jennifer A. Doudna and Samuel H. Sternberg. They are the canonical writers of this account and were at the absolute forefront of that 2012 breakthrough.
Yes. And while we focus on their firsthand account, it is so crucial to recognize Emmanuelle Charpentier's role.
Right.
She was Doudna's central collaborator in developing the CRISPR-Cas9 system. And together, Doudna and Charpentier shared the 2020 Nobel Prize in Chemistry for that breakthrough.
Wow, yeah.
Now, Charpentier is not a writer of this specific book, but her scientific contribution to the platform is, it's foundational.
Yeah, and I think it's also important to correct a common misconception here, one that the authors themselves are actually very careful to address in the book. People sometimes talk as if Jennifer Doudna created CRISPR all by herself in a vacuum.
Right. Like she just invented it on a Tuesday.
Exactly, yeah.
Yeah.
But this was a massive, sprawling web of parallel research. You had microbiologists, geneticists, biochemists, people studying obscure salt marsh microbes in Spain,
Yep.
or scientists at dairy companies trying to figure out why their yogurt cultures were dying from viral infections. All of them contributed to this foundational knowledge over decades. All of that collective incremental science is what made the 2012 reprogramming possible.
Science is inherently a relay race.
Mm.
A landmark discovery always relies on the quiet, often unglamorous work of hundreds of researchers who came before. Without the scientists who sequenced those weird repeating patterns in bacterial DNA in the 1980s and 90s, the 2012 breakthrough just never happens.
Let's talk about how this actually works in practice, because this is where the public understanding gets a bit fuzzy, I think.
Sure.
If this tool is essentially a reprogrammed bacterial defense system, how exactly does it edit human DNA?
Well, to understand that, we really need to look at the trio of components that make this machinery work.
Okay.
So first, you have target recognition. Second, you have the guide RNA, which scientists can now program in a lab to match a specific 20-letter sequence of DNA. And third, you have the Cas9 enzyme, which does the physical cutting.
Right, the cutter.
Exactly. But there's a vital catch that often gets left out of the, simplified explanations. Cas9 will only cut if it finds a specific short DNA sequence right next to the target.
Oh, really?
Yeah, it's known as a PAM sequence.
A PAM sequence. What is that, like a biological password?
More like a zip code, actually. So imagine you have a massive library, and that is the human genome, right? 3 billion letters long.
Okay, big library.
The guide RNA is the street address you were looking for. But the Cas9 enzyme is kind of like a mail carrier who absolutely refuses to deliver the package unless the zip code is verified first.
Oh, I see.
The PAM sequence is that zip code. It's a built-in safety lock, ensuring the enzyme doesn't just cut randomly everywhere. It finds a partial match to the address. It has to find the address and the zip code right next to it.
That makes total sense. But when I read about this in the news, people always use the phrase genetic scissors.
Oh, all the time.
Yeah, I see it in almost every headline about CRISPR. You program the scissors, they go in, they snip out the bad gene, and you're cured.
It is a highly effective metaphor for public communication, but it is dangerously incomplete. And frankly, it gives people a false sense of security.
How so?
Well, when you hear scissors, you think the cutting is the edit. You think it's a clean, finished process like, cutting a coupon out of a newspaper. But cutting the DNA is only the very beginning of the editing process.
Wait, really?
Yeah, Cas9 just breaks the DNA strands. It does not put them back together. It has literally no mechanism for repairing what it just broke.
Here's where it gets really interesting, because we aren't the ones doing the actual editing at all, are we?
No, we aren't.
The cell's own natural repair mechanism is doing the heavy lifting. CRISPR basically just creates a massive emergency inside the nucleus, and the cell rushes in to fix it.
Precisely. We're just triggering the alarm. And the cell's response to that alarm is where things can get incredibly chaotic.
Chaos in the cell. Great.
Yeah, because when a double-strand break occurs in DNA, the cell basically panics. A broken DNA strand can be lethal, so the cell tries to rejoin the severed ends as fast as humanly possible.
Okay.
This default emergency pathway is called non-homologous end joining.
Non-homologous end joining.
Right. It's quick, but it's incredibly sloppy. Think of it like hastily taping a torn manuscript page back together. The cell almost always accidentally inserts or deletes a few DNA letters in the process.
Which ruins the word at that location.
Exactly. For scientists, this sloppy repair can actually be highly useful if their only goal is to simply disrupt or, knock out a disease-causing gene. If you mutate the gene enough, it just stops functioning entirely.
But what if you don't just want to break a bad gene? What if you want to fix it? Like actually rewriting a specific typo in a genetic sentence so that a protein functions correctly again?
Right, well, that is much harder. To do that, you rely on a completely different repair pathway called template-directed change, or homology-directed repair.
Okay, homology-directed repair.
Yeah. So along with the CRISPR-Cas9 machinery, you supply the cell with a customized DNA template that contains the correct sequence. The hope is that the cell will use your supplied template as a blueprint to patch the break cleanly.
But I'm guessing it doesn't always want to do that.
Not at all. The reality is that the sloppy, quick repair pathway is the cell's default. Competing repair pathways are constantly fighting each other. The cell might completely ignore your beautifully engineered template and just mash the broken ends back together anyway.
Which naturally corrects a massive misconception out there. People think CRISPR produces perfectly predictable edits, like a word processor. You just hit backspace, type in the new code, and hit save. But it's not a magic wand. It is highly probabilistic.
It is entirely probabilistic. You are banking on cellular mechanisms that you do not fully control. And that brings us to the scientific limitations and risks discussed in the book.
Right.
First, there's off-target activity. Remember the zip code analogy?
Yeah, the PAM sequence.
Right. Sometimes the guide RNA might find a sequence that is almost a perfect match, maybe just one letter off, and the Cas9 enzyme cuts there by mistake. You could be trying to cure a blood disorder and accidentally disrupt a healthy, vital gene that suppresses tumors.
That is terrifying. You go in to fix one problem and accidentally create cancer somewhere else in the genome.
It is a very serious concern, which is why there is so much relentless focus in the field right now on improving enzyme fidelity.
Yeah, making it more accurate.
Exactly. But it gets more complicated. Because even if Cas9 cuts exactly at the intended on-target site, like no off-target mistakes at all, you can still get unexpected changes. The cellular repair process we just talked about might cause massive unintended deletions or rearrange the DNA sequence right at the target site.
So even a perfect cut can lead to a disastrous repair.
Yes. Furthermore, you have the problem of mosaicism.
Okay, what does mosaicism mean for an actual patient? I've heard that term thrown around.
Right, so it means that if you deliver CRISPR to a population of 1 million cells, not every single cell is going to receive the exact same edit. You might end up with a mosaic mixture.
Ah, like a patchwork.
Exactly. Some cells are perfectly edited using your template. Some cells remain completely unedited, and some cells have those sloppy, unintended knockout edits. In a lip patient, that heterogeneity completely shapes whether the therapy is actually effective or safe.
Let's talk about those living patients, because keeping all of that unpredictability in mind, it is wild to think we are actually using this tool right now. This takes us firmly into somatic editing. And let's draw a line in the sand here, because this is where a lot of ethical confusion starts. What exactly is somatic editing?
We have to define this very clearly because it changes the entire ethical landscape. Somatic editing means altering specific, non-reproductive cells in an existing person.
Okay, so not embryos.
Right, not embryos. We are talking about blood cells, liver cells, muscle cells, retinal cells in the eye. You are treating the patient sitting in front of you. Whatever changes you make to their DNA will die with them.
Got it.
The edits will absolutely not be passed on to their children or future descendants.
Okay, so if we are trying to fix a somatic cell, we have to physically get the CRISPR machinery into that specific tissue. The book talks about this as a delivery bottleneck, because it is one thing to drop CRISPR into a petri dish full of cells in a sterile lab. It's entirely different to navigate the human body.
Oh, delivery is arguably the decisive constraint of genome editing right now. There are generally two overarching strategies, ex vivo and in vivo.
Ex vivo meaning outside the body?
Yes, ex vivo means out of the living. You take cells out of the patient's body, edit them in a highly controlled laboratory environment, check to see if the edit actually worked and is safe, and then you transplant those cells back into the patient.
Okay, and in vivo?
In vivo means within the living. This is much harder. You inject the CRISPR components directly into the patient's bloodstream or tissue, often using viral vectors or lipid nanoparticles, and you just have to hope it navigates to the right organ, enters the correct cells, and makes the edit safely without hitting off-target tissues.
Wow. Ex vivo sounds vastly more controllable, even if it's, logistically complicated.
It is.
And that actually brings us to a massive post-publication milestone that we have to talk about. The book we're discussing came out in 2017, when a lot of clinical application was still theoretical promise. But in December 2023, The FDA actually approved the first CRISPR-based therapy called Casgevy for sickle cell disease.
That approval is a true watershed moment in human history. It proved that somatic editing could successfully move from theoretical promise to a regulated, highly effective treatment for an inherited blood disorder.
And sickle cell is just awful.
It is devastating. It is caused by a single genetic mutation, one wrong letter out of billions, that makes red blood cells warp into a rigid sickle shape. These malformed cells clog blood vessels, causing excruciating pain crises, progressive organ damage, and significantly shortened lifespans.
But the way Casgevy works isn't by just going in and fixing that sickle mutation directly, right? I was reading about the mechanics of this, and the biological elegance of the approach is just stunning.
It is incredibly sophisticated. Rather than trying to correct the sickle mutation, which, as we discussed with template-directed repair, is difficult to do cleanly and efficiently, Casgevy uses the sloppy, easier repair pathway to its advantage.
Oh, really? How does it do that?
It disrupts a regulatory region associated with a gene called BCL11A.
What does BCL11A do?
Normally, BCL11A acts like a biological switch. When we are in the womb, we produce a special kind of hemoglobin called fetal hemoglobin, which is incredibly efficient at carrying oxygen. Shortly after we are born, the BCL11A switch flips, turning off fetal hemoglobin and turning on adult hemoglobin.
Okay, I follow.
For someone with sickle cell, that adult hemoglobin is defective. So by using CRISPR to break that BCL11A switch, the therapy effectively forces the patient's body to revert to its infant state and start producing fetal hemoglobin again.
Oh.
Yeah, this healthy fetal hemoglobin dilutes the defective adult hemoglobin, preventing the red blood cells from sickling.
That is absolutely brilliant. It's like finding a biological backdoor to bypass the disease entirely. But I do want to push back on the reality of this treatment for the actual patients.
Please do.
Because molecular elegance in a lab or even regulatory approval from the FDA does not automatically equal accessibility. To get this ex vivo treatment, think about what a patient actually has to go through. First, they have their stem cells collected. Then they have to undergo myeloablative conditioning.
Yes, and we need to explain what that means. Myeloablative conditioning is essentially intense, highly toxic chemotherapy designed to completely wipe out the patient's existing bone marrow.
That sounds horrific.
It is. You have to clear the soil, so to speak, to make room for the newly edited stem cells to engraft and grow.
Right. It is a grueling, dangerous process. Patients face prolonged hospitalization, severe risk of life-threatening infections because they temporarily have no immune system at all, and potential permanent infertility from the chemotherapy itself. Not to mention the staggering financial reality. We are talking about millions of dollars per patient. The infrastructure required to extract cells, edit them in a clean room, administer chemo, and monitor a patient in isolation is immense.
Which naturally corrects a huge, persistent misconception out there.
Yeah.
The fact that we have one approved effective therapy absolutely does not prove that every CRISPR application is suddenly safe, easy, or universally accessible. The people suffering the most from sickle cell disease globally, particularly in sub-Saharan Africa, are in regions with the least access to this kind of multi-million-dollar, highly specialized medical infrastructure.
It's a sobering reality check. The science is practically miraculous, but the societal execution is deeply flawed. Now, if somatic editing is about treating the patient in front of you, despite all those hurdles we just mentioned, what happens when we point these precise tools not at a liver or a blood cell, but at embryos, eggs, or sperm?
When you do that, you cross a massive biological and ethical line. You are moving from somatic editing to heritable or germline editing. And it is so crucial to correct the misconception that somatic and heritable editing are ethically equivalent.
Right.
They are worlds apart.
I want to dig into that because some people might argue, if it cures disease, what's the difference? Why are they in completely different ethical universes?
Because of consent, irreversibility, and the scope of affected persons.
Okay, unpack that.
With somatic editing, an adult patient with sickle cell can weigh the risks of chemotherapy against the benefits of the edit, and they can give informed consent. And if something goes wrong, the tragedy ends with them.
Right.
With heritable editing, you are altering an embryo, a future person who obviously cannot consent to being biologically engineered. You are introducing an intentional, experimental biological change with deeply uncertain effects. And crucially, that edit becomes part of the human gene pool.
Forever.
Yes, it will be passed down to their children and their children's children forever.
And this isn't just a philosophical debate about a distant future anymore, is it? We've already crossed this line. In 2018, the scientific community was absolutely rocked by the announcement that a researcher, He-Jiang Cui, had actually edited human embryos that resulted in live births.
The 2018 embryo case proved in real time that scientific capability can effortlessly outrun global consensus. Even when there are widespread ethical condemnations, international moratoriums, and strict prohibitions in place, if the tools are relatively cheap and accessible, which CRISPR is, someone, somewhere, might cross the line.
Yeah, that was a huge wake-up call.
It completely transformed the conversation from an abstract ethical thought experiment at bioethics conferences into an urgent, terrifying enforcement problem.
So let's look at the arguments people make for crossing that line, because the motivations aren't always villainous. Usually the argument starts with prevention.
Right.
Imagine you are a parent sitting in a genetic counselor's office. You and your partner know you carry a devastating, lethal monogenic disease, a disease caused by a single faulty gene, like Tay-Sachs or Huntington's, and you want to ensure your child doesn't suffer from it.
Prevention of severe, untreatable suffering is undeniably the strongest, most sympathetic argument for heritable editing. But even in those heartbreaking cases, the authors and the broader scientific community have to ask about reproductive alternatives.
What kind of alternatives?
Well, in the vast majority of cases, those couples can use in vitro fertilization combined with pre-implantation genetic testing. They can screen the embryos in a lab and select one that doesn't carry the disease, entirely avoiding the need to edit DNA and risk off-target mutations.
So you don't even need CRISPR for that.
Usually, no.
But even if we agreed that editing for severe disease was acceptable, the slippery slope isn't just a cliche here. The line between preventing a severe disease and enhancing a human being is incredibly blurry.
Oh, very blurry.
Once you establish that it's biologically safe and legally acceptable to edit an embryo to prevent cystic fibrosis, the very next person in line asks to edit an embryo to increase muscle mass, or select eye color, or boost intelligence.
That ventures into polygenic enhancement. And here is a critical scientific reality check that bursts a lot of science fiction bubbles. Most traits people consider enhancements, like intelligence or height, are highly complex.
Meaning they aren't just one gene.
Exactly. They are polygenic. That means they are influenced by thousands of different genes interacting in cascading networks that we do not fully understand, coupled with massive environmental influences.
Right, which completely shatters the misconception that genes are like simple toggle switches that determine every human trait. We are not simple machines where one line of code equals one exact output.
No, we are far too messy for that.
If you try to tweak 50 different genes in an embryo trying to make a child taller or faster, you have no idea what else you are breaking.
Yeah.
You might inadvertently increase their risk for severe autoimmune diseases or neurological disorders.
Precisely. We simply do not possess the biological mastery to safely engineer complex traits, even if we universally agreed it was ethically acceptable to try.
Which we haven't.
No. This raises an important question, though, regarding the rhetoric we use when we talk about eliminating disease from the human gene pool. How does the disabled community view this relentless drive toward genetic perfection?
I am so glad we are covering this because the book dives into this, and it cites a perspective that gets completely sidelined when everyone is just, marveling at the technology.
The discussion often lacks profound nuance. Disability advocates argue in the literature that there is a deep, valid concern regarding the rhetoric of gene editing. When society talks enthusiastically about editing out conditions like deafness or dwarfism, it can inherently devalue the lives of people currently living with those conditions.
Yeah, that makes a lot of sense.
They point out a critical distinction between the medical model of disability, which views the person as broken and needing to be fixed, and the social model, which argues that people are often disabled by a society that refuses to accommodate human diversity.
It's entirely possible to support a patient's individual right to seek relief from painful medical suffering, while also recognizing that society has a terrible track record of deciding which types of humans are valuable,
Yes.
which casts a very long, very dark historical shadow over this whole field.
It casts the shadow of eugenics.
Mm-hmm.
Historically, eugenics movements in the 20th century used flawed claims about heredity to justify horrific state coercion, racism, immigration restrictions, and forced sterilization.
Yeah, horrific track record.
Now, contemporary gene editing in a modern fertility clinic looks very different from state-sponsored eugenics. But the authors caution that we have to be intensely vigilant. If access to germline editing is unequal, if it costs millions of dollars, and it is driven by intense market pressures and societal norms of perfection, We could easily create new, biologically reinforced social hierarchies.
It wouldn't require a totalitarian government mandating anything. It would just require wealthy parents feeling a competitive obligation to optimize their children so they don't fall behind.
Exactly. A consumer-driven eugenics driven by inequality and advertising.
Wow, that is a heavy reality to sit with. But as we pivot here, we have to remember that CRISPR's power to control evolution doesn't stop at human biology. We are talking about the entire biosphere.
Oh yeah, everything with DNA.
Think about the food in your fridge right now. What does this technology mean for agriculture and animals?
For agriculture, the promise is significant and moving incredibly fast. Researchers are editing crops to be more drought-resistant, or editing wheat to resist fungal diseases, or creating mushrooms that don't brown as quickly to reduce food waste.
That sounds great on paper.
It does. As climate change radically disrupts global food security, these tools are vital. But again, molecular success in a controlled greenhouse doesn't automatically mean ecological success in a complex field environment. And the book points out that we have to consider the socioeconomic reality of corporate seed ownership.
Right, because if a massive agrochemical company patents the drought-resistant wheat, who actually controls the food supply?
Exactly.
Do independent farmers maintain autonomy, or are they locked into restrictive intellectual property agreements where they have to buy licenses every season just to survive?
It's a critical tension. And when it comes to livestock, the ethical calculus gets even more complicated. You could use CRISPR to edit a pig to be immune to a devastating respiratory virus, like PRS.
PRS.
Yeah, porcine reproductive and respiratory syndrome. On the surface, that sounds like a massive win for animal welfare.
But if that disease resistance just allows massive industrial factory farms to cram those animals into even worse, more densely crowded conditions because they won't get sick and die as easily, you haven't actually improved animal welfare. You've just optimized industrial suffering for higher profit margins.
Precisely. Scientific feasibility does not answer whether an application is ethically justified. You have to look at the entire system the animal lives within, not just its DNA.
Speaking of complex systems, we need to talk about gene drives. I read the sections about this application for mosquitoes, and it absolutely blew my mind.
It's wild stuff.
The analogy I keep coming back to is that a gene drive is like putting a biological steering wheel on an entire species.
That is a very apt analogy. To understand why it's so radical, you have to look at normal inheritance. Normally, thanks to Mendelian genetics, a specific genetic trait has a 50-50 chance of being passed from a parent to their offspring.
Right, basic biology.
A CRISPR gene drive is engineered to completely cheat that system. It actively cuts the opposing chromosome and copies itself over, ensuring that almost 100% of offspring inherit the targeted trait.
So mechanically, you could design a gene drive that makes female mosquitoes sterile. You release a few of these engineered mosquitoes into the wild in a targeted area, they mate with wild mosquitoes, and instead of the trait diluting over time, it forces that sterilizing trait through the entire population exponentially.
Yes, very quickly.
Within a few generations, the population crashes.
Yes. The public health appeal of this is enormous. You could potentially eradicate malaria or dengue fever, diseases that kill hundreds of thousands of children every year, by simply wiping out the mosquito vector.
Which would be an incredible achievement for human health.
It would.
Yeah.
But the ecological risks are immense. Ecological systems are infinitely complex food webs. If you release a gene drive, you have to understand that national borders are porous. A mosquito doesn't care about passport control. It will not stay confined to one country.
And once it is released into the wild, it's out there. Reversal is extraordinarily difficult, if it's even possible at all.
It exemplifies the core terror of this technology. Local technical power, a few scientists in a well-funded lab, can create irreversible collective consequences for the entire planet.
Which is the perfect reason we have to discuss the absolute necessity of global governance, with applications ranging from curing sickle cell disease in a hospital to editing the food supply to literally rewriting wild ecosystems. Who is actually in charge of keeping this safe?
The short answer is it's complicated. There is no single global department of CRISPR.
Right.
We have a fragmented system of layered oversight. Individual laboratories manage internal biosafety protocols, universities have ethical review boards, national regulators like the FDA or the EMA evaluate clinical products, and international bodies try to set broad norms.
The book discusses how the scientific community has tried to self-regulate, drawing parallels to the famous Asylumar conference in the 1970s for recombinant DNA.
Yes, where scientists got together to establish safety guidelines before the government stepped in.
Right. But in 2021, the World Health Organization released a comprehensive set of recommendations specifically for human genome editing.
Yes, the WHO recommendations focused heavily on creating a systemic approach to governance. It isn't just about banning things. It's about establishing meaningful consent protocols, mandating global research registries so we actually know who's doing what in their labs, ensuring independent oversight, and creating mechanisms to monitor accountability.
They also specifically addressed medical tourism, right?
Yes, they specifically addressed the threat of medical tourism, where desperate patients might travel to countries with lax regulations to get unproven, dangerous therapies.
But the tension here is that technologies always evolve faster than laws. How do you govern a molecular tool that is changing and improving by the week?
You need adaptive governance. The rules cannot be so static that they become completely obsolete in a year, but they also can't be so flexible that they bow to intense commercial acceleration and profit motives.
Right, you need a balance.
Some boundaries, like this strict prohibition on heritable embryo editing, need to remain firm and enforceable, while the technical safety standards for somatic editing can adapt as our clinical evidence improves.
Yeah, that makes sense.
If we connect this to the bigger picture, governance isn't just about safety, it is fundamentally about equity. We have to look at intellectual property and patent disputes.
Oh, the patents.
The foundational patents for the CRISPR-Cas9 system have been tied up in massive, bitter legal battles for years between major research institutions.
Right, because whoever owns the patent controls the platform.
Exactly. How those foundational patents are licensed dictates who gets to do basic research, which startups can afford to develop new therapies, and ultimately which patients get access to the cures and at what price. Affordability and access are as much a part of governance as safety regulations.
So what does this all mean? We started this massive journey by asking how we can use this unprecedented power to reduce human suffering without causing irreversible harm to our biology or our society.
It means that the ability to rewrite DNA creates possibilities, not permission. Technical precision, the ability to cut a DNA strand exactly where we want, does not provide moral precision.
Yeah.
As the authors powerfully argue, our scientific capability must be joined to profound humility, a steadfast commitment to global justice, and a long-term responsibility for the ecosystems and future generations we share this planet with.
Before we leave you with our final thought, we need to state clearly that this has been an independent Cosmora HQ book overview. This is not an audiobook, an authorized adaptation, an endorsement, or a substitute for lawful access to the published work. We highly encourage lawful access to the book to explore these incredible ideas in their full depth.
And we invite listeners to subscribe to Cosmora HQ, share your perspective in the comments, and share this deep dive with an intellectually curious friend.
As we wrap up, I want to leave you with something to mull over. We talked briefly about how intellectual property and patents dictate who can use CRISPR. Think about the long-term implications of that. We are essentially allowing the foundational tools of planetary evolution to be governed by corporate patent margins and market monopolies rather than global democratic consensus. If humanity can rewrite the code of life, who should decide which changes are allowed?